Okay, hello everyone. My name is Cam. I'm a 40 medical student at the University of Oxford. So today I'm going to be delivering this talk on metabolism, specifically lipid, glucose, and amino acid metabolism, with a few other bits thrown in, like ketone metabolism as well. This is obviously quite a broad topic, so I've got quite a few slides. I'll try and kind of get through them quite quickly, and then people can view over the material in their own time if they want to go over things. And there's a few other slides that won't be shown in the presentation just because they're kind of extra topics like diabetes, for example, pathophysiology, and cholesterol synthesis, which we just don't have time to do. So, but you'll get the slide shared to you so you can have a look at those in your own time. So first of all, just starting off, I'm going to do it out of order of what's written on the first slide. So I'm going to do glucose first because I think that's the most important one, and then we'll do lipids and then amino acid metabolism as well. So first of all, just kind of this is quite a good summary slide of organ metabolism. So various different types of tissue in the body, various different organ systems, and what the kind of main substrates that they use for metabolism, and what they get their kind of main, what's their main source of energy is. What this is really highlighting, and what I've actually explicitly highlighted here, is just that every tissue pretty much will use glucose to varying extents. Some tissues are incredibly reliant on glucose, for example, the brain, and also erythrocytes, red blood cells, because they lack mitochondria. They can't do oxidative phosphorylation, which is obviously a separate topic, so we won't go into that. And then there are other tissues like the heart, for example, that are less reliant on glucose and are more reliant on free fatty acids, which we'll go over as well. So first, we'll go over glucose. So glucose is the predominant kind of metabolic substrate for most tissues in the body. I'm sure you may already be aware of kind of the process of glucose metabolism, but we're going to go through it systematically in the next few slides. So the first thing is just kind of starting from quite a macro point of view is what's regulating the uptake of glucose. So after you eat a meal, for example, a carbohydrate-rich meal, those carbohydrates, you swallow them, they go into your stomach, they get broken down by various enzymes, which will break them down into more simple monosaccharides, disaccharides, those kind of individual sugar units. And one of the major ones of those is glucose. That glucose will then be absorbed from your gastrointestinal system into your bloodstream, and then it's in your bloodstream, and then the cells of your tissues have to be able to access that. So there's two hormones that are predominantly responsible for regulating that process, and they have antagonistic effects on one another. So insulin is the hormone responsible for increasing and stimulating glucose uptake in cells. And not only is it kind of bringing glucose into cells for metabolism, it's also bringing glucose into other tissues like the liver, for example, which can then use it to convert into glycogen through glycogenesis, which we'll talk about later, as a process of kind of storing excess glucose from food, which you can then use later in anaerobic conditions, for example. And glucagon has the opposite effect. So if your blood glucose levels are low, your cells, particularly those in your brain, which are heavily reliant on glucose, are deprived. Your body and specifically your pancreatic alpha cells in the islets Langerhans will secrete glucagon, which have the effect of stimulating glucose release from the liver through two processes, which we'll talk about later, glycogenolysis and glucogenesis. So this is a nice little graphic of it. Okay. This is a bit more specifically about the kind of how glucose gets into cells. So there's different transport proteins on the cell membrane, and these are present in different tissues. Glucose uptake is through facilitated diffusion. And yeah, as I said, there's different tissues with, there's different glucose transporters in different tissues which have different affinities. There's this concept of Km, which is kind of a constant which is kind of inversely proportional to your affinity. And there's a definition of it there, but you can read that yourself. So if you've got if a glucose transporter has a low Km, it means it has a high affinity for glucose. So you've got GLUT3 transporters in the brain, for example, and in neurons, which have very low Km. They have very high glucose affinity because, as we've said, the brain's very heavily reliant on glucose. So any glucose that's in the blood, the brain's going to try and get hold of that as quickly and efficiently as possible. The only one of these which is insulin dependent is GLUT4. I think that's quite an important fact. GLUT4 is present in skeletal muscle, in adipose tissue in the heart, as it says there. So your muscles, particularly during exercise, have a very high metabolic rate. They've got very limited supplies of their own glycogen for glycogenolysis. So they have a very high need for glucose, but this is dependent on insulin. And there's also these additional ones, SGLT1 and SGLT2. These SGLT2 is quite important clinically. This is present in the kidney, and there are drugs that are now used to treat high blood pressure and for diabetics, which are SGLT2 inhibitors, which increase glucose loss through the urine by basically inhibiting this process of glucose uptake in the kidney. Okay, so glycolysis. Glycolysis is the first part of glucose metabolism. So the purpose of this is it's kind of capturing some initial energy as ATP from glucose, and is also producing these reducing agents, NADH, which can then go on to into mitochondria and participate in the kind of oxidative phosphorylation in mitochondria to produce additional ATP. So that's the kind of main point of it. The overall process is that you're converting a 6-carbon glucose molecule into 2 3-carbon molecules of, well, it says here glyceraldehyde, 3-phosphate, and dehydroxyacetone phosphates. And then you're going through further processes to produce molecules of pyruvates, which can then enter the citric acid cycle, which we won't really talk about in this one because I think that's more related to kind of mitochondrial metabolism and oxidative phosphorylation. The big point here is that glycolysis is not oxygen dependent. Glycolysis is the process that will happen both in anaerobic and aerobic conditions, but the ATP yield per glucose molecule is much lower in glycolysis, which I think will come on to the next slide. The only other things on this is there's kind of 3 key regulatory steps in glycolysis, the kind of irreversible steps, which are catalyzed by these 3 enzymes written at the bottom of the page. Hexokinase, which catalyzes the first step. This is quite important because the conversion of glucose to glucose 6-phosphate then traps glucose within the cell. So this is quite an important process so that you keep the glucose that you've taken up from the bloodstream, facilitated by insulin concentration. You then manage to store that glucose 6-phosphate inside the cell. Something mentioned here is there's an isozyme of hexokinase, called glucokinase, which has a much lower affinity for glucose and therefore is only active at very high intracellular concentrations. And this is present in tissues like the liver, where it kind of acts as a glucose sensor. Phosphofructokinase catalyzes the 3rd step of glycolysis. This is the rate limiting step. And as you can see, with all of these enzymes, they're inhibited by some of the products that come after them. So it's a kind of negative feedback loop for glycolysis. And then the final one is pyruvate kinase, which catalyzes that final step. So insulin and glucose will also decrease and increase the activity of all 3 of these. Insulin will kind of increase the activity of these enzymes to increase glycolysis, and glucagon will decrease it. So how many ATPs are generated from glycolysis? So glycolysis overall is only going to produce 2 ATP per glucose molecule. So it's not very efficient, which is why you can't, I mean, other than the fact that you're producing lactate, it's why you can't indefinitely respire anaerobically. You can't sprint forever. And then from aerobic respiration, you've obviously got the additional bits. You've got the link reaction, which converts pyruvate to acetyl-CoA, and the Krebs cycle, the citric acid cycle, which is talked about separately. And the ATP yield from aerobic respiration is anywhere between 30 and 32 ATP molecules. And it will depend on how much NADH or FADH2, which are produced from both in glycolysis and from the citric acid cycle, FADH2 specifically, depending on which of these shuttles you're using to get those coenzymes into the mitochondrial matrix. So this is something about the role of pyruvate dehydrogenase. So this is the one that catalyzes the link reaction. It links glycolysis to the TCA, citric acid cycle. It's irreversible, and it's regulated by these enzymes mentioned here as well. PD phosphatase catalyzes the reverse process of the final step here. Yes, from pyruvate to phosphoenolpyruvate. Yeah, you can read over these data in your own time. There's something on the syllabus about galactose and fructose and how these link into glucose metabolism. So you can read it here. So fructose can enter into the glycolytic pathway directly, and it can be used for glycogenesis and lipogenesis. Galactose is converted to glucose, on the other hand, before it enters. And then there's a bit here. What if these pathways don't work? Well, this links quite nicely to some pathophysiology. So you can get two conditions. So you can get galactosemia, where you have a mutation in these enzymes. Well, either this one, type 1, I'm not going to try and pronounce, it's a long word, or type 2, where you've got a mutation in galactokinase. So it's a loss of function mutation. They're autosomal recessively inherited, leads to an accumulation of galactose in the blood. And much like any of these genetic abnormalities, you get these kind of symptoms in very young children. So lethargy, jaundice, slow growth, that kind of thing. And then, okay, and then hereditary fructose intolerance. So here you have the first process is fine, fructose to fructose 1-phosphates, and then you have a mutation in this enzyme, fructose 1-phosphate aldolase, which causes an accumulation of fructose 1-phosphates, and this will kill your liver cells. So that's more about that. Okay, let's keep going. Both of these are treatable by dietary restriction. Okay, so you're missing an enzyme that helps utilize galactose and fructose. So the way of treating it is just to avoid those sugars. Okay, this is just a slide about isozymes. I'm just going to go through this quite quickly. The point is that isozymes have kind of similar overall functions. So hexokinase and glucokinase, as I mentioned, is a good example. They both have the same functions. They catalyze that initial process of glycolysis from glucose to glucose 6-phosphate, but they're present in different tissues. And then there's a few other examples here which you can go over in your own time. Okay, so the pentose phosphate pathway. So what is this? Okay, well, this is a bit about where it is and the point of it. I don't know why we've got all these animations, but there you go. So the pentose phosphate pathway has a couple of functions. So one of the main ones is it's responsible for producing ribose 5-phosphates, which is a nucleotide which can be used for nucleic acid synthesis, so DNA and RNA. It's also used to reduce glutathione, which I'm not really going to talk about in this. And it's also used in fatty acid synthesis. The pentose phosphate pathway has these two stages, oxidative and non-oxidative, and the regulation. So it's regulated, and its rate limiting step is the first enzyme in the pathway, glucose 6-phosphate dehydrogenase. So glucose 6-phosphate is coming from glycolysis. So this is kind of an offshoot of the glycolytic pathway. So what's the relevance of these products? So as I kind of briefly mentioned, ribose or ribulose 5-phosphates, I think these are two separate molecules, but essentially ribulose 5-phosphate has just been converted to ribose 5-phosphate. This is a precursor for, this should say purines, not pyridines, purines and pyrimidines for DNA and RNA. And also producing some glycolytic intermediates as seen there. And another major role of it is it generates NADPH, which is a cofactor for a whole load of different processes in the body, like fatty acid biosynthesis, glutathione recycling, as I mentioned, nitric oxide synthesis, which has important activities in the lungs, for example. I think that's all there is on that slide. Yeah, okay. So in anaerobic respiration, what happens to all the NADH generated? So if you remember from the glycolysis pathway, you're overall producing NADH. And now what should happen to that NADH is it should then go into the mitochondria and then be used in oxidative phosphorylation. But what happens if you don't have oxygen? So therefore you stop that pathway. What happens to the NADH? So as it says here, it accumulates because the electron transport chain doesn't allow it to be oxidized. So you get a shortage of NAD plus, so reduced NAD. And then eventually you won't be able to undertake glycolysis. You get a buildup of lactate because this enzyme, lactate dehydrogenase, kicks in to regenerate NADH. So it allows you to cycle through this pathway, which is great for a while until you then end up with way too much lactates. So what do? You do something called the Cori cycle. So this is a way of recycling lactates produced from anaerobic respiration in a whole variety of tissues. Specifically, this is quite a simplified diagram explaining the production of lactate from muscle. This pathway essentially is a way of recycling it. So that lactate then gets taken through the bloodstream back to the liver, where it's converted back to glucose through gluconeogenesis in the liver. And this pathway can then cycle through. And as you can see, the amount of ATP, being produced from glycolysis is 2, as we mentioned, but the gluconeogenic pathway requires 6 ATP. So there's a net loss of 4 ATP per glucose molecule metabolized through this pathway, which is, again, another reason why you can't just indefinitely respire anaerobically, because you will rapidly become depleted of your ATP molecules. Again, that's just a bit of detail. Okay, so we'll go over glucose storage, and specifically the kind of metabolism of glycogen, which is the main storage molecule for glucose in the body. So the structure of glycogen is a branched polysaccharide consisting of glucose molecules bound by 1 to 4 glycosidic bonds, and every 4 or so you have 1 to 6 glycosidic bonds, which allows for this branching structure. And there's 2 places that your body predominantly stores glycogen. One is the liver, which is where most of your most of your body's glycogen is stored, and in your skeletal muscle as well. That in your liver can be released into your bloodstream because of the presence of a particular enzyme, which we'll come on to later. Muscles, on the other hand, lack this enzyme, which prevents the glucose from being released into the bloodstream. But yeah, so this branch structure allows it to kind of be held quite compactly in tissues until it needs to be used. So glycogenesis, this occurs in the fed state. So when you've got lots of insulin and lots of glucose around, the liver cells are going to take up and the muscle cells are going to be taking up that glucose in the bloodstream. And then it goes through this pathway written out here and to form these kind of glycogen branches. It starts from a kind of central polypeptide molecule called glycogenin. And you have both these kind of, you're producing the 1 to 4 glycosidic bonds and the 1 to 6 glycosidic bonds as well, every 8 glucose molecules or so, which allows for this nice branch structure. So then glycogenolysis is the reverse process. So you've got basically what happens is you've got a variety of phosphorylase and debranching enzymes. And what this will do is it will it will bind onto one of these side chains, and it will essentially relocate it to the kind of main chain. And the purpose of this is that it makes it more efficient for this enzyme 1,6-glucosidase to kind of individually cut off these glucose monomers, which can then be released into the bloodstream for neighboring tissues, or indeed in the muscle. So this is the point here that I mentioned before. Muscle lacks glucose 6-phosphorylase, which is this last process. So I think it's just another name really for 1,6-glucosidase. So glucose 6-phosphorylase, for the same reason that we mentioned earlier about glycolysis, the first process of glycolysis being that process of converting glucose to glucose 6-phosphate, this traps the glucose into the cell. So if you don't have the reverse enzyme, which in this case is glucose 6-phosphorylase, you cannot then release that glucose back into the bloodstream for other tissues to use, which is why the glycogen in muscle can only be used by the muscle itself, whereas the liver glycogen can be released into the bloodstream. Okay, so a bit about hormone regulation. So glucagon adrenaline promotes glycogenolysis and prevent glycogenesis through activation of protein kinase A. I'm not really going to go into too much detail about these molecules. The main point is that glucagon and adrenaline will the function is to increase your blood glucose levels. So they will want to increase the amount of release of glucose into the bloodstream. So they'll increase the lytic pathway that's breaking down that glycogen. And insulin will have the opposite effect through activation of this enzyme, phosphoprotein phosphatase. Okay, so gluconeogenesis. Gluconeogenesis is a very interesting process, essentially de novo synthesis of glucose from nothing, or not from nothing, but from an alternative molecule that isn't in any way related to glucose potentially. This occurs in the liver and the kidneys. Majority of it occurs in the liver. So, for example, and this will occur when you're fasting, because you have to maintain a certain level of glucose in your bloodstream. And in order for tissues like your brain to have sufficient energy to kind of undertake respiration. So as you can see here, in short fasting, the majority of the gluconeogenesis is done in the liver. This is kind of short fasting when you're asleep. And then if you've got prolonged fasting, so you haven't eaten for a few days, a couple of days, more of this burden for gluconeogenesis will shift to the kidneys, but the majority of it is still done by the liver. So you might ask now, okay, well, is gluconeogenesis just glycolysis in reverse? And the answer is not quite, because if you remember, there were those three enzymes, hexokinase, phosphofructokinase, and pyruvate kinase, which are irreversible in glycolysis. So you have to bypass those pathways with an alternative enzyme, and that's kind of nicely written out on this diagram over here. So you've got glucose 6-phosphatase, which we mentioned, which the muscle doesn't have, fructose 1,6-bisphosphatase, and two enzyme complexes that get around the pyruvate kinase pathway, which produces oxaloacetate in the middle, which can enter the TCA cycle as one of those substrates. So yeah, you need quite a high ATP burden to do gluconeogenesis. So it's not the most efficient pathway in the long term. So you can't kind of continue doing this indefinitely. Substrates for gluconeogenesis. There's a few different molecules, as I mentioned, that can be used for gluconeogenesis. Glycerol, which comes from the hydrolysis of triacylglycerides. Fats. Lactate produced from anaerobic respiration and supplied to muscles through the Cori cycle. Supplied to liver by the Cori cycle for gluconeogenesis. And oxaloacetate, which we mentioned, and also some amino acids. So these are some of the ones mentioned: alanine, cysteine, glycine, serine, threonine, tryptophan, and glutamine. I think that's glutamine. But the ones that are highlighted are the kind of predominant ones, are alanine and glutamine. These are also important for certain kind of pathways like the Cahill cycle, which we'll come on to later. So how is futile cycling avoided? So you can imagine if you have glycolysis and gluconeogenesis, particularly in liver cells occurring in the kind of the same tissues with almost exactly the same enzymes, bar a few, then why don't you have just constantly making glucose, getting rid of it, making glucose, getting rid of it? And the reason is because you've got reciprocal regulation. So the signals that are inhibiting one pathway will activate the other. So this is and insulin glucagon is a prime example of that. And as you can see here, glucagon is a primary driver of gluconeogenesis, and it's inhibiting glycolysis, unlike insulin, which does the reverse. So there you go. That's a nice little summary of it there. Okay, so this is just a brief bit about experimental evidence of glucose as fuel. I'm not really going to go over this too much. You can read about these in your own time, but there's kind of two big studies that were done on this that kind of use as evidence for using glucose as fuel. The main points out of this was that the first one, and I'm not going to try and pronounce the name because I will butcher it, but this first study showed that oxygen uptake was increased when you added carbohydrates. So it suggested that this was an oxidative metabolic pathway. And then Krebs and Johnson in 1937 found that what their study showed was that the concentration of citrate wasn't really changing overall. So this suggested that if you're adding substrates, it's essentially just going round and round. It's a cyclical pathway. Okay, why can't fatty acids be used to produce glucose? This is just a point that was raised, so I think in the syllabus, so I thought it was quite important. The reason is because the transition reaction converts fatty acids to acetyl-CoA. And we'll get onto that with beta oxidation. And acetyl-CoA is that produced from the link reaction. Acetyl-CoA cannot be converted back to pyruvate. As we mentioned before, the link reaction is irreversible. And it also can't just go through the TCA cycle to become oxaloacetate because you need an additional carbon-containing molecule to enter the cycle, because overall in the TCA cycle, you're losing CO2 molecules, two CO2 molecules. So essentially, the carbon molecules from acetyl-CoA will disappear. So you can't just keep going round and round. Fates of pyruvate and acetyl-CoA, they're just written here. I'll let you guys read over that in your own time. Glycogen storage disorders. There are a few different ones. I think the kind of key ones are von Gierke disease, Corey disease, and McArdle disease. It shows you the deficient enzymes, and if you look at the glycolytic and glycogenic pathways, you can see where those enzymes, which process those enzymes are responsible for. But the key one is glucose 6-phosphatase catalyzes that process of glucose 6-phosphate to glucose to enter the bloodstream. McArdle disease is just muscle glycogen phosphorylase, so it will have symptoms unique to the muscle itself, unlike these other ones, and the glycogen debranching enzyme in Corey disease. So essentially, you can't debranch, which is quite an important process in the first step, because you need to move the branches on the glycogen to the kind of main branch in order to allow these other enzymes like glucose 6-phosphatase to release those individual glucose molecules. And then inborn errors of metabolism. So these are just genetic defects that affect metabolism. And again, this is a nice summary slide, which I'll let you guys read over in your own time, grouped together into intoxication disorders, energy metabolism disorders, and storage diseases as well. Okay, so we're going to move on to lipids. That was the biggest one, glucose. So I think these others should be a bit shorter. So advantages and disadvantages of lipids as an energy source. So the advantages are they're very abundant in food sources. I mean, I'm sure you're all aware of how easy it is to get hold of high-fat foods. They're incredibly abundant in food sources, okay? And they're super energy dense. So per gram, it's over double the amount of energy in calories compared to glucose. It's also very efficient in terms of storage because lipids are fundamentally hydrophobic. So they all kind of coagulate together, which allows you to store them in kind of a nice compact space. And you essentially have unlimited capacity because you can continue building up your adipocytes, excuse me, in your tissues, and essentially just carry more and more and more fats almost indefinitely until you're putting stress on your other organs, which is what happens in obesity, for example. The disadvantages of lipids, they're very slow to break down, which we'll go over. It's a very long, laborious process to break down lipids, which is why it's not the kind of predominant energy source for the majority of cells in the body. It's just a slow process. There's many steps. It's only aerobic. You won't get this in anaerobic respiration because you need oxygen. And for the same reason that the fact that it's hydrophobic is great for storage, it also makes it difficult to transport it between adipose tissues and target tissues. It needs to be bound to a protein like albumin, for example. Okay. So organ use. So you can see here that, as I said, in the fed state, most tissues are using glucose, with the exception of cardiac muscle. In the fasting state, on the other hand, alternative pathways are used because even though the process is slow, if you don't have any glucose around, you haven't really got much choice. And fatty acids, as we mentioned, very energy dense, so you might as well go through the beta oxidative pathway and get hold of that sweet ATP. Yeah, this is just a bit more about kind of organ use. Okay, lipid sources. The majority of dietary fat, about 90%, is triacylglycerol, which is a glycerol molecule backbone with 3 esterified fatty acids that are added to it. These esterified fatty acids can be classified based on the number of double bonds. So saturated fats have no double bonds, and then you have monounsaturated, polyunsaturated, sorry. The only other thing on here is there are 2 essential fatty acids. So linoleic acid and alpha linoleic acid, very similar structures. Essential being that your body cannot produce these endogenously. They have to be gained from dietary sources. And if you are deficient in either of these, you'll have some of these symptoms similar to the kind of symptoms you get from other metabolic disorders we mentioned earlier. So skin problems, stunted growth, poor wound healing, increased infection risk, that kind of thing. Okay, so fat metabolism. The first step after you've eaten something and it's gone into your stomach and it's digested by some lipases, there's also some lipases in your mouth and your saliva, is assimilation. So this is this process of getting those, what can be potentially really complex fats into your bloodstream for use by tissues. So this will happen with emulsification with bile, which is produced in the liver. It's stored in the gallbladder and then released into the proximal part of your intestine, in your duodenum, where it will help emulsify those fats so that they can then be absorbed into the bloodstream through your enterocytes. So and then which pathway they go down depends on the size. So short and medium chain fatty acids can just go directly into the bloodstream from your enterocytes. Longer fatty acids are reformed into those triacylglycerols, triacylglycerides, and then they're packaged as something called chylomicrons. And then these are released into the lymphatic system. So they bypass the liver. And then these chylomicrons, which are kind of made up of, well, here we go. There you go. There's a nice little summary of what happens. And this is your chylomicron. So it's a big, messy molecule of protein, phospholipids, cholesterol. And what will happen is these will circulate in the lymphatics, and then kind of lipids from that, specific fatty acids, will be kind of broken off by enzymes and then taken up by tissues before circulating back to the liver. So here you go. So this is this kind of first process. These are your chylomicrons down here, and where it says 1. And then a lipoprotein lipases, which we'll come on to in tissues, will then take off those fatty acids, and then the chylomicron remnants will return to the liver, where they will then be kind of repackaged. Okay. And then the HDL pathway. So HDL, high density lipoprotein. We haven't really talked about the LDL and LDL yet, but we'll come on to that. So your kind of low density lipoprotein, this is what people think when they think of cholesterol. Your LDL, VLDL, HDL. Your VLDLs are your kind of very low density, big, loads of kind of fatty acids, cholesterol. And then eventually those kind of bits of fatty acids are going to be picked off those by lipoprotein lipases on cells. And this will get smaller and smaller. And then your HDL is the kind of goes around and scavenges up those free cholesterol and fatty acids. Lipoprotein metabolism. I don't think I'm going to go over this. This is something you can read in your own time. But there you go. Yeah. So LDL is very cholesterol rich. It's one of the things that when people talk about bad cholesterol, that's what they're talking about. Okay, lipoprotein lipase. So these are enzymes on the surface of endothelial cells, and they hydrolyze the triacylglycerols from chylomicrons for tissue uptake. And then what happens to the chylomicrons? They circulate back to the liver and they're recycled as VLDL, which then circulate in the bloodstream. This is where also your short and medium chain fatty acids absorbed from enterocytes go directly to the liver via the portal circulation from the gut. So the chylomicrons just take a slightly longer pathway, basically. So what happens if you're deficient? If you're LPL deficient, then you can get a whole host of different conditions. And these will cause elevated levels of circulating triglycerides, which can cause things like abdominal pain and pancreatitis. Treatments for these conditions are reducing your fat intake, essentially. This is a nice little diagram. So these are the kind of symptoms you can get, and this is what a blood sample would look like in someone who had something like hyperchylomicronemia or any of these other LPL deficient diseases like hypercholesterolemia as well, which we'll talk about later. Okay, lipid metabolism in the fed state. I'll just go over this really quickly. So in the fed state, you've got loads of fatty acids, you've got loads of glucose. Insulin-dependent GLUT4 is going to activate, take up glucose, and you have LPL, which on certain tissues, which is going to be taking up fatty acids from your chylomicrons, from your VLDL, and insulin promotes all of these enzymes. So you're getting fatty acid uptake, getting glucose uptake, and it's also going to inhibit this enzyme, hormone-sensitive lipase, which is found in certain tissues which break down stored fats in, for example, in adipocytes. So you do that in the fasting state when you need those fatty acids to be accessed. Yeah, and then the fasted state, essentially the opposite process is happening. And this is activated by hormones like adrenaline, which is the catecholamine, growth hormone, and cortisol. Now, but why? Why would you increase those molecules? So in the fasting state, your hypothalamus in your brain is going to detect the low glucose, and this will send signals via the anterior pituitary glands to release this hormone ACTH, which then acts on the adrenal glands to increase cortisol and adrenaline, and also via the sympathetic nervous system through noradrenaline. And this is essentially going to have all the actions that we've talked about in terms of increasing glycogenolysis, increasing glucagon secretion, increasing blood glucose, increasing hormone-sensitive lipase activity to increase fatty acids in the blood to basically give as much energy as possible in a low glucose environment. Okay, so this is the carnitine shuttle. The carnitine shuttle is responsible for the uptake of fatty acids from the cytosol. So once they've got into the cell, into the mitochondrial matrix for beta oxidation. So this is the process of breaking down fatty acid chains into acetyl-CoA, which can then enter the TCA cycle and be used for ATP production and energy. So it's essentially you have these kind of protein complexes, or you have carnitine which binds to acyl-CoA. You've got these two enzyme complexes, CPT1 and CPT2, and it's essentially acting as a transporter across the inner mitochondrial membrane. A notable point here is that short and medium chains, so if it's less than 8 carbons, you don't need carnitine because these can just enter the mitochondria nice and easily. But if you've got really long ones, if you've got, well, greater than 8, but we're talking like 20 carbons, then you need the carnitine shuttle to get those into the matrix. Regulation of the carnitine shuttle. So malonyl-CoA will inhibit CPT1. So this is the first process of converting acetyl-CoA to produce stored lipids, essentially. So this prevents beta oxidation. So what the kind of result of this is that you're stopping lipid biogenesis, which happens in the liver and happens in adipocytes, and beta oxidation, which is lipolysis, occurring at the same time. Because there's no point just making something and breaking something at the same time, right? So how is this affected by carbohydrate metabolism? So excess acetyl-CoA, which is produced from glycolysis, will leave the mitochondria as citrate. And the point of this is this will then activate this enzyme acetyl-CoA carboxylase, which will increase lipid biogenesis, which is why if you eat lots and lots of high carbohydrate food and you don't do any exercise, you will probably put on fats because it will store that, because there's only a limited amount of glycogen that could be stored, but there's an unlimited amount of fats that could be stored. Okay, so this is beta oxidation. I'm not really going to go over this. You can go over this in your own time. The main point is that through each cycle, you're losing 2 carbons at a time through this kind of set of processes. You're also producing these coenzymes like NADH and FADH2, which can then enter the oxidative phosphorylation pathway in mitochondria. And you're giving off acetyl-CoA as you go. And you'll basically just keep going around down the fatty acid chain, giving off acetyl-CoA molecules as you go. This is just something about kind of non-esterified fatty acid levels in different tissues and in the blood, depending on hormone levels. It's essentially just rehashing what we've already said. So I'll skip over that. Lipogenesis occurs in the liver, occurs in adipocytes. So as we kind of mentioned, so the excess acetyl-CoA will leave the mitochondria as citrates, and then distinct enzymes called fatty acid synthase, which we've got a brief slide on, will then basically build up those fatty acid chains. And it's essentially a reverse process. And there's your malonyl-CoA, which we mentioned before. This is the fatty acid synthase. We're just going to briefly go over this, but yeah, there's a big complex of lots of different enzymes, essentially. This is quite a complex slide, again, going through the process. You can review that in your own time. This is comparing beta oxidation and lipogenesis. Again, go over this in your own time because I realize I'm running out of time. So I'm going to go on to proteins and ketones. Okay, I'll go over this briefly. So energy usage during exercise. So during exercise, your skeletal muscle is contracting, you're using a lot of energy, you have increased ATP demand. So you need to activate both pathways that are oxidizing carbohydrates and fats at the same time. And there's various different things, intensity, duration, type of exercise, that are going to affect the mechanisms through which the extra energy is supplied. So in skeletal muscles, you can get hold of glucose in the blood, so you can get hold of glycogen in the muscle itself, liver glycogen, and also this thing called creatine phosphate, which is quite small. There's a very small amount of it, but it's used to resynthesize ATP by creatine kinase to replenish depleted ATP levels. So this is how it changes. So in fasted low intensity exercise, you can get most of your energy from beta oxidation because you've got the time, essentially. If you're doing quite intense exercise, you've got more beta oxidation, which is going to be happening, but you've also got increased use of glucose as well. And then in intense exercise, it's kind of going to flip the other way. So you're going to get most of your energy from carbohydrate oxidation, which is much faster and much more efficient, but you have less ATP production per molecule. And then if it gets really intense and you can't get enough oxygen, you're then going to go through anaerobic glycolysis, which is going to generate your ATP, but you're going to get a buildup of lactate, and essentially you're going to have that loss of overall ATP through the Cori cycle, so you can't do that indefinitely. Energy expenditure in the heart. So most of this, again, is coming from fatty acids. If you have during exercise, because your heart obviously is incredibly reliant on enough energy, you don't want that to kind of stop working. Lipolysis that's happening in adipose tissue and in the liver is going to increase circulating lipid levels and fatty acid levels, sorry, which your heart can then get hold of. Okay, a few pathologies. So MCAD deficiency is a deficiency in medium chain acyl-CoA dehydrogenase, which is one of the enzymes involved in this beta oxidation pathway. So you basically get you can't break down these fatty acids, you can't get hold of the energy from them. And this will result in things like severe hypoglycemia and even death from fasting, because essentially in the fasting state, you just cannot get enough energy from lipolysis. So not good, basically. And it's surprisingly common, actually. In terms of treatments, I think it's essentially you just have to avoid fasting. You have to keep eating, and not keep eating all the time, but you have to make sure that you don't go long periods of time without fasting, sorry, and decrease your fat intake as well, so that you don't have as many of these medium and long chain fatty acids hanging around. Okay, familial hypercholesterolemia, also surprisingly common. This is where you have there's different proteins that can be mutated: ApoB100, LDL receptor, and PCSK9. But LDL receptor is an important one. So it basically means that you just have increased LDL in the blood and you get all kinds of strange symptoms. You get atherosclerosis, you get young people in their 30s developing heart attacks and atherosclerosis from this. You also get quite a significant clinical sign, which is tendon xanthomas, which are little kind of fatty deposits, usually seen on the backs of the hands, around the eyes. Treatment for this is statins, reduce your circulating cholesterol. Carnitine deficiencies, I'm not super, I don't know, I'm not an expert on this. But again, this is something you can read in your own time. And Jamaican vomiting sickness, I thought this was quite niche. I'd actually never heard of this before I made these slides. But apparently, so you have these toxins from the ackee fruit, which I guess presumably was originally found in Jamaica. And these toxins will inhibit the same enzyme, these kind of medium and short chain acyl-CoA dehydrogenases. So you basically prevent beta oxidation from happening. So what will happen is you'll get a buildup of these fatty acids, you'll get hypoglycemia, and then you'll get these awful symptoms. Leads to non-ketotic hypoglycemia. Treatment for this, you need to replace the glucose. You give a glucose infusion, you give fluid electrolyte replacement, and you wait until the person clears it from their system and gets better. Okay, ketone metabolism. Okay, I'm going to need to go over this quite quickly, but so ketogenesis occurs when you don't have enough oxygen. When you don't have enough glucose. So your fatty acids are converted into a glucose replacement. So what happens is that beta oxidation, if you've got lots of beta oxidation, you'll produce way too much acetyl-CoA. It will saturate the TCA cycle. You'll get a depletion of intermediates, so it will just completely slow down for the same reason that, we mentioned earlier, that acetyl-CoA has to come in and you need other intermediates as well. Ketogenesis is a kind of temporary solution. It occurs really in conditions like type 1 diabetes, where you just cannot access glucose in the bloodstream, and that fatty acid buildup will produce ketone bodies, which cells can use for energy. So yeah, and it's because insulin would normally inhibit hormone sensitive lipase, but without insulin, you don't get that. So you get fatty acid breakdown. So adipocyte tissue breakdown and ketogenesis. This is just quite a complex slide, basically outlining the diagram. Ketone bodies refer to acetoacetate, acetone, and D3 beta hydroxybutyrate. And these can then be used to generate NADH and for oxidative phosphorylation. So essentially a glucose replacement. But they're toxic to the body. This is just a little bit at the bottom about regulation. I'm not really going to go over that. Okay, ketogenic versus glucogenic amino acids. So a glucogenic amino acid is one that can be converted to glucose via gluconeogenesis. Ketogenic amino acids can be converted to ketone bodies. Some do both. Actually, most of them do both. This diagram nicely outlines, I think, pretty much all the amino acids and whether they're ketogenic, glucogenic, or both. Amino acid metabolism. So most amino acids are obtained from dietary proteins, especially essential amino acids, i.e. the ones that you cannot produce endogenously. Usually the more complex ones or the cyclical ones, like phenylalanine and tryptophan, for example. Some of it comes from your body recycling its own protein. And also you can get synthesis of amino acids from metabolic precursors as well. So all of these pathways all interlink into one another. So where do the amino acids go? They can be used for all kinds of structures. The nitrogen from them is disposed of through the urea cycle, which we'll go over. The carbon skeletons can be used for processes like the TCA cycle, gluconeogenesis, ketogenesis, as we mentioned, and others can be used for things like neurotransmitters and nucleotides and porphyrin, which is the base for hemoglobin. Transamination, desamination. This is the process of kind of the slow process of kind of removing kind of nitrogen from these molecules, and you're basically moving the amine group, the H3N group, to other molecules to essentially remove them from your body, to then remove the nitrogen through the urea cycle. Again, I'll let you guys go over this in your own time. Regulation of fate of deaminated amino acids. So as we said, the carbon skeletons can be used for a whole load of different processes, including gluconeogenesis and ketogenesis, but the nitrogen has to be removed because ammonium is toxic to the body. This can build up in liver failure, for example, and in advanced liver failure, you can get hepatic encephalopathy, where you get toxicity in the brain due to a buildup of ammonium. The urea cycle. Okay, so this is essentially the process of getting rid of that nitrogen. And so this is the ammonium arriving here at the top, and it gets converted to urea. Urea is then excreted in the urine. Brilliant. Yeah, I think that's the main thing. The control of the urea cycle acutely, enzyme regulation. This first enzyme is the rate limiting step. And in chronic, it can be upregulated. For example, if you've got high protein diet and you've got high need to remove that nitrogen. Right, amino acid metabolism. This is quite quick. Food's broken down by these enzymes, go into the liver. Hormones will increase those insulin will increase those anabolic processes, and those amino acids can be used for protein synthesis. Excess are broken down as keto acids, enter the TCA cycle or lipogenesis. Fastest state, low blood glucose promotes glucagon release. This will increase glucose release and ketogenesis, and the carbon skeletons can be used as gluconeogenic substrates. You'll also get large breakdown of amino acids sent to the liver for gluconeogenesis via the Cahill cycle in the first week of fasting, so you still have glucose accessible to the brain. And then proteolysis will occur later, and you'll get ketone body reduction. So this is the Cahill cycle, which we mentioned briefly. This is a cycling between glutamates and alpha-ketoglutarate and alanine as well. I think that's something I should leave for you to read in your own time. Okay, these are the last couple of slides, which is good. I've actually done okay for timing. So pathologies of amino acid metabolism. So phenylketonuria is the most common error of amino acid metabolism. So this is where you've got a autosomal recessive loss of function of the enzyme phenylalanine hydroxylase, which prevents the conversion of phenylalanine to tyrosine, which is used for as a kind of initial substrate for a variety of molecules, including things like dopamine and melanin. Symptoms of these, so the symptoms will present in children, young children, and they'll have things like intellectual disability due to low levels of neurotransmitters, hypopigmentation, and in terms of treatments, you have to decrease phenylalanine from the diet. So they have to eat quite specific things, and you must supplement tyrosine. It's quite difficult to do because phenylalanine is a common dietary protein. But this is a condition that's screened for in newborns. Maple syrup urine disease, great name. Not very pleasant for the person with it, but you have a disorder in that you have a mutation, loss of function mutation in this enzyme, branched chain ketoacid dehydrogenase. And you essentially get a build-up of branched chain amino acids, which is quite a few different amino acids. And the reason for the name is that essentially your urine apparently smells like maple syrup due to a buildup of isoleucine. I don't think this is a very common disorder, really, but it's important from a kind of kind of basic science understanding point of view. Yeah, it's not good. It causes neurological problems called ketoacidosis, and you have dietary restriction lifelong as a treatment. And then the last slide I've got is alkaptonuria. This is a deficiency of this enzyme, opogandisic acid oxidase. So you get a buildup of this molecule HGA. The kind of hallmark of this, you'll have arthritis, you'll have black urine because of a oxidation of the urine. And those are kind of the key things I think to consider with this one. It's not fatal, unlike the previous two, but you can get debilitating arthritis from it. And finally, the treatment for this is restriction of both phenylalanine and tyrosine. I think that's it. Yes, thank you very much for listening. I did actually manage to get through that in an hour. There are additional slides which have been shared on diabetes and cholesterol, as I mentioned, and you can go over the slides in a bit more detail if you please. Thanks for listening. Please, if you could fill out the feedback form. I know I was kind of rushed and I went through it quite quickly, but if you wouldn't mind giving some feedback, it'd be really, really helpful. It's helpful for me to know, what I do well and what I don't, and what I can improve on, and also if there needs to be changes to the slides, which can be done as well. So there's the QR code and there's this form as well. If you've got any questions about any of this, there are some email details there, both my email and that for OSME itself. But yeah, thanks very much, everybody, and enjoy the rest of your day.